Good morning students, I am Shomajit sir. Today I am here to deliver my lecture on topic artificial methods of propagation in plants as well as on tissue culture. So this is from the chapter how do organisms reproduce. So watch the entire lecture to get basics out of it. So without any delay, let's start with the topic. So let's start with the topic artificial methods of reproduction in plants and tissue culture. So you can see the figure, rows cutting from day 1 to day 18 you can see. So we'll see this one by one. You can see the layering methods and all. So we'll see this one by one. Now in artificial propagation of plant, the process of growing many plants from one plant, alright, the process of growing many plants from one plant by man-made methods are called artificial propagation in plants. So we know that the process of growing many plants from one plant by man-made method is called artificial propagation in plant. First one is cutting. What is cutting? Let's see. A small part of the plant, which is what is cutting? Cutting is a process in which a small part of the plant which is removed by making a cut with a sharp knife is called cutting. Alright. So whenever a small part of plant is removed by making a cut with a sharp knife is called cutting, and cutting may be a piece of stem, roots, or even a leaf. Now you can see the cutting diagram. This is day 1 and this day 18th. On 18th day you can see it's the leaf has grown. Alright. So grow rows from cutting. So you can grow rows from cutting. By cutting method you can grow rows. Now moving to layering, we'll see the different types of you can see the layering methods also. We'll see, you see very nicely given, one plant is there and the one branch is twisted. All right, few portion of the plant is dipped inside the soil and the next portion is upright, the shoot is upright. So later on it will be cut and separate plant will be formed from one plant. Now what is layering? In this method, a branch of the plant is pulled towards the ground and a part of it is covered with moist soil, leaving the tip of the branch exposed above the ground. So after some time, new roots develop from the part of a branch buried in the soil. As I have said, the branch is then cut off from the parent plant and the part of the branch which has developed roots grow to become a new plant, just like parent plant. Example we do in jasmine plant or chameli is propagated or produced by layering. Right, layering method is produced. You can see different types of layering are there: air layering, simple layering, serpentine layer, tip layer, trench layer. All right, mound or stool layer. Next is the grafting. What is grafting? All right, one is scion and its stalk will come, like two different plants are cut. All right, the stem is cut in a angled way, slanting way, so that it gain more surface area, and the top part of the plant of top part of the shoot that is from the other plant is called scion, and the bottom part of the plant that is attached with the root is called stalk. We'll see this one by one, and then it will be tied up to form a one plant body. So what is a grafting? All right, grafting is a method in which the cut steps of two different plants, one with roots and other without roots, are joined together in such a way that two stems join and grow as a single plant. So first one is the cut stem of the plant or tree having roots and fixed in soil is called stalk. All right, what is stalk? The cut portion of the or the cutted stem of the plant having roots fixed in a soil is called stalk, and the cut stem of another plant without roots is called scion. Now you can see the advantages of artificial vegetative propagation. One is seedless plant. So seedless plant can be produced by artificial propagation. Next point is many plants can be grown from just one parent plant by artificial propagation. Third is the plants grown by vegetative propagation usually need less attention in their early years than the plants grown from seed. Okay. And next advantage is that the new plant produced by artificial vegetative propagation will be exactly like the parent plant. So in artificial vegetative propagation, the new plant produced from the plant will be exactly similar. All right. So no genetic variations will be seen. And many plants can be grown from just one plant. All right. And for this method, it require vegetative method. It require less attention. You can give less attention to the plant. Now moving to tissue culture. What is a tissue culture? All right. So tissue culture is in vitro cultivation of plant cells or tissue under aseptic and controlled environmental conditions. All right, under aseptic and controlled environmental conditions. So in liquid or on semi-solid, well-defined nutrient medium for the production of primary and secondary metabolite or to regenerate a plant. All right. So small callus is taken that are totipotent in nature. All right. So in a slide, in a petri dish or in a test tube, that tissue will be there that is highly totipotent in nature, that has active power of cell division, so that they can grow roots and shoots from there. And then we can apply hormones like auxin, cytokinin, ethylene, gibberellin acid. All right, different hormones can be used. If I apply hormones on that particular plant, in particular that callus tissue, then if I apply auxins on that callus tissue that have highly defined cell division, all right, and they has a capacity to divide a cell. So if I apply auxin hormone or give auxin hormone on that particular tissue, it will give root formation, it will yield root formation. And if I give gibberellin, all right, then it will do shoot formation. So you can see the applications of tissue culture. Tissue culture is used to conserve the rare species in the forest. A plant breeder may use tissue culture to screen cells rather than plants. Larger scale growth of plant cells in liquid culture as a source of secondary products like recombinant proteins used as biopharmaceuticals. So these are the applications of tissue culture. Now we can see a tissue culture in a test tube. Callus tissue is taken from there, the auxins, gibberellins are applied. You can see roots are formed and stems are formed, all right, shoots are formed in an artificial medium. It has been done, and later on, after the plant is grown, you can put it on a natural environment. You can place it on a top first, and then you can place it on a soil later. You can see in a test tube how the plants are grown from a callus tissue, all right, that has a totipotent or active power of cell division, that can do rapid cell division. Now what are the advantages of plant tissue culture? First is availability of raw materials, second is variation in supplies and qualities, third is parent rate, fourth is easy purification of the compound, fifth is modification of chemical structure, sixth is disease free and desired product, and seventh is crop improvement. Now student, these are the homework questions. First is define the following with one example of each. One is cutting, layering, grafting. Second is what are the different advantages of artificial vegetative propagation in plants. Third is define tissue culture and also mention its advantages. Fourth is mention also the application of tissue culture. Thank you student. This was all about the entire video lecture of artificial propagation in plants and tissue culture. Hope you have liked the video and understood. So stay at home, stay safe. I'm Shomaji sir signing off for today, and again we'll meet in the next class. Thank you students.